Hi all, welcome back. The last topic I want to talk about is how that impulse works, moving from a one neuron to either a muscle or to another neuron, okay, and we call that synapses. So synapses is the site where the neuron transmit that nerve impulse to another neuron, or it can be to a muscle, to the motor plate of a muscle, or to a gland. Okay. So a presynaptic neuron is the neuron that's sending the impulse. A postsynaptic neuron receives the impulse. Okay, so prior to synapsis, presynaptic, that neuron sending the impulse, postsynaptic, after synapsis, okay, that neuron receives it. Okay, so there are two separate neurons, okay, two separate kind of clefts. Okay, you have a synaptic transmission, um, okay, and this is the process by which a synaptic neuron sends the impulse to the postsynaptic neuron. Okay? And this is going to be similar to what we were talking about when we were talking about how muscles work, where you have a action potential move down the axon. And that opens up calcium gates. Calcium flows in. Calcium changes the permeability of the membrane, which allows for the neurotransmitter to move across the membrane into the synaptic cleft and then into the uh, neuron or open the gates so material can move into the other neuron. Normally that's opening up sodium gates so sodium can travel in, resulting in another action potential. And we'll cover this. We'll go through each one of these steps. Okay, so here you have the presynaptic neuron. Okay, so this is the neuron that is going to be sending the signal. So in both cases, we have two presynaptic neurons attached to a postsynaptic neuron. Okay, and they're not technically attached, they're just really close to each other. There's a little space called the synaptic cleft. And here you can see you can see that the neurotransmitters are being released into the synaptic cleft. So you have this action potential moving down the axon that changes the permeability, changes the cell membrane here to allow for that neurotransmitter to move into the synaptic cleft, which changes the permeability of the cell body in this case, okay, this neuron, which then will excite an action potential to move down this axon. Okay? So here's a little better look at that synaptic vessel, okay, at that synaptic knob. Okay, so if we dive down here, that impulse is going to open up channels. Okay, so that that action potential moving down into the synaptic knob is going to open up channels. Typically, these are calcium channels. Calcium will then flow in. Okay, that changes the membrane, the axon membrane, and it allows for these synaptic vessels, which hold neurotransmitters, to move to the outer edge, be incorporated into the membrane. Okay, so you can see here, their membranes merge and the neurotransmitter is released into the synaptic cleft. That then will bind up to some kind of receptor on the nerve that it's communicating with, the postsynaptic nerve, or a muscle. It doesn't really matter. Same kind of premise works in both. Okay, and they bind up, and that changes this membrane's composition, which normally it's opening up sodium channels so sodium can flow in. Okay, and here you can just see kind of what it looks like when you do a cross section of that synaptic knob. So you have that cleft, okay? You have the postsynaptic membrane of another nerve. Okay? You have synaptic vessels in here. You have mitochondria for energy, because the system needs energy in order for it to occur. And then you have cell, like other cell tissue that's associated with it, okay, and axon moving down. So that synaptic transmission, okay, is that transmission of a nerve impulse from one neuron to another, okay? The release of the neurotransmitter across the synaptic cleft, we already talked about that. That's going to change the composition and open up receptors on the membrane. Okay. It really depends on what the neurotransmitter is, and we'll come back and we'll talk about this, but you have some neurotransmitters which are excitatory, so they're going to excite a response, which means that they're going to carry or cause action potentials to be carried from one to another, okay? And then you have repressor or neurotransmitters that repress the process and stop action potentials from occurring. So they can either open up ion channels or close ion channels, or do both, open some and close others. Okay? And it's really just a matter of, we're just talking about ions, things like sodium, potassium, calcium, moving across membranes. They're changing the potential of the membrane. They're changing the charge inside the membrane versus outside the membrane, and that allows for that action potential to occur. So excitatory neurotransmitters are going to increase the permeability of sodium ions. So it causes those sodium channels to open up, and sodium can now travel inside the cell, okay, whether that be a muscle cell or another nerve cell, okay. And then you have inhibitory neurotransmitters that's going to block, or it really doesn't open the sodium channels. Instead, it might open up other channels like chloride channels to have it move in the opposite direction. Okay. So two real types of potential, okay? One we call EPSP, or excitatory postsynaptic potential, okay? And this is when the sodium channels are open, okay, which causes depolarization and causes an action potential to be carried, okay? Inhibitory postsynaptic potential, or IPSP, okay, is really kind of the opposite effect, but it's not that all the sodium channels are closed. Yes, some of them are closed, okay, but it's more like you open up potassium channels or open up chloride channels, so you have the opposite effect. Okay? You go into hyperpolarization, okay, or hyperpolarized membrane, which means that potassium is going to leave the axon, okay, which changes the action potential, and it doesn't allow that action potential to occur, or at least it's less likely. Okay, in both cases, it's just a matter of likelihood, because it really depends on how much stimuli you have in the system. Okay, and that's really where EPSP and IPSP come into play, is they're both working collectively. Okay, so it's basically a summation of the two. So you have things that are going to excite, right, and open up sodium channels, and then you're going to have inhibitors that are going to open up potassium channels or chloride channels. And if there's a greater amount of excitatory, then action potential probably will occur, okay, or the likelihood is much greater. If there's a greater amount of inhibitory, the action potential is probably not going to occur. Okay? So it's really based on the probability of an action potential. Okay? Remember, we're trying to get to that negative 55. So if we're exciting, okay, but we're not exciting at quite enough rate, okay, or there's more inhibition in the system, then we won't reach the threshold. Okay? If there's a far more amount than inhibition, okay, it'll just continuously be negative. Okay, so a net inhibitory effect does not generate an action potential. Okay, that's pretty logical, right? Net inhibitory rate. Okay, a net excitatory rate probably will. Okay, eventually. In this case, it looks like it's going to drop off because it's not quite enough. But the summation of all the inputs occur, and that has to make it to kind of that trigger zone. Remember that depolarization zone. Once you get to that point, then the action potential will be pushed down the axon. Now, neurotransmitters. This gets really, really complex, but we're just gonna barely touch the surface. Okay, so the material that is held in the synaptic vessel, okay, in that synaptic knob, is some type of chemical. Okay, normally some type of protein. It's typically a protein-based chemical, and there's lots of them. Okay, we haven't discovered all of them yet, and the way that they work is there's receptors on the other side for that given chemical. Now, the interesting piece with that is you can have some neurons that use multiple neurotransmitters, and then some neurons are specific to maybe one neurotransmitter. If they're specific to one neurotransmitter, they'll typically carry the name of that neurotransmitter. Okay, but often it's kind of difficult to know what neurotransmitter is being released unless you study that specific neuron. Acetylcholine is a very, very common neurotransmitter because it's stimulating muscle contraction. Right? And we know that there's roughly 600 muscles, skeletal muscles, more than that, okay? All of which can be stimulated by acetylcholine. So neurotransmitters can be monoamines, amino acids, peptides. Okay, again, normally they have a root in protein-based material. Okay, you can have neurotransmitters that are produced in the rough ER or the cytoplasm. Remember, we talked about this. They're really produced where ribosomes are at. Okay, and so the rough ER where ribosomes are attached, or the cytoplasm where ribosomes tend to float around. Okay, and we already talked about how that works. Once the action potential reaches the synaptic knob, okay, the neurotransmitters can be released through what we call exocytosis. So endocytosis is bringing in things. Exocytosis is merging your membrane and releasing material out. Another word for that is vessel trafficking. Okay, and that's the process by which we just recycle membranes. So these vessels that occur, these little packets that have these neurotransmitters, these proteins or amino acids in them, okay, they merge with the cell membrane and they release the material out. Okay, and that's what we call vessel trafficking. Okay, and so you're just recycling the membranes. Okay. It works the same way when things get engulfed and brought in. Okay. Endocytosis, the membrane flexes, it bends in, material comes in and it engulfs it and encapsulates it in a membrane and brings it in, and now that forms a new vessel and whatnot. Okay. So a couple other neurotransmitters, just real quick, we can throw some out there. Acetylcholine, again, a very, very common neurotransmitter, both in the central nervous system and the peripheral nervous system. Norepinephrine, okay, this is also a really common neurotransmitter in both central nervous and peripheral nervous system. Dopamine, most of us know about dopamine. I've heard of probably these 3 or 4, maybe even 5, all of them. But dopamine's another one. Okay, this is a typical neurotransmitter that kind of gives us a sense of well-being. So it stimulates kind of a has a kind of a calming effect in a way in the central nervous system. It's kind of limited in the peripheral nervous system, but it can be excited or inhibited depending on where it's at. Serotonin, okay, again, this is what leads to sleepiness. Okay, histamines, okay, this is what often results in reactions to allergens and things like that, and promotes alertness, especially of the immune system. Okay, we'll come back and talk about histamine. So again, how's this work? Just to recover, reshoot kind of these 6 steps. Action potential is passed along the axon. We know that. Gets to the synaptic knob. Okay, the synaptic knob membrane becomes more permeable to calcium ions. Okay, that permeability allows for the synaptic vessels to fuse with the synaptic membrane. Those vessels, when they fuse, they're going to release a neurotransmitter, one of the 100 plus that are known. Okay, those neurotransmitters will make it into the synaptic cleft. They react with the receptor on whatever cell membrane is next to it, whether that be muscle, nerve, glandular, okay? And that provides a change in the next cell's membrane, okay? And so it can be through endocytosis, it could bring that material in. It could just be a reaction that changes the channels on a membrane so other things can come in. There's all kinds of situations that can happen. All right, neuropeptides. All right, so these are neurons that are synthesized in the brain and the brain and the spinal cord. Okay, they both can be transmitters. Okay, or they can be modulators. Okay, so a modulator is not an inhibitor. Okay, a modulator typically will not block receptors necessarily, but might block the release of a transmitter for a limited amount of time. Okay, and so the modulators are less invasive, I'd say, than the than the inhibitors. Okay, and so we'll look at this here shortly, okay, when we talk about some things. So enkephalins, okay, these are pain sensations, so they relieve the pain sensation, okay. Beta endorphins, this thing like opioids are beta endorphins, okay, they relieve pain sensation also, okay, tend to be longer lasting. Substance P is what's conducting pain impulses. So enkephalins and endorphins inhibit the release of substance P. Okay, so that pain impulse is controlled by both of these. Okay, so these are modulators at time. Okay, they modulate the pain release or the transmission of pain impulses. Okay, but they're not detonating it to where it doesn't occur any longer. Okay? They're just limiting the pain impulse and inhibiting the release of the substance P for a while. Okay, so let's look at an example of something that's very, very common and can be a huge problem, especially in the United States, and that's opioids or opiates. Um, Opiates or opioid drugs like morphine, heroin, codeine, opium, okay, they are potential for painkillers because they are endorphins. So they stop the, they inhibit the production of pain, okay, or at least the signal for some types of pain, okay. Um, and so there's other opiates that are naturally occurring in the human body, like ankylosis, okay? And there are some that just occur naturally as endorphins in the body. And then, of course, modern medicine, uh, we've made artificial types of endorphins, which will react and stop the production of pain, okay, or the signal of our pain receptors. Okay, so they're very useful to relieve severe pain, okay? But they're very addictive from the perspective of the experience that individuals get from the opiate is not just a relief of pain, but it's a relief, or it's a production of euphoria, or production of kind of this happiness, this calmness, etc., that is also associated with the opiate drugs. So you can have lots of situations when you have withdrawal symptoms. First of all, you have increased pain, okay, because now you have nothing that is relieving the pain, okay, and you have increased pain, and that euphoria is gone. And so now the other piece is often the self-made, self-producing endorphins, okay, stop producing when you have opiate drugs that are being put into the body. Right? So you get the natural endorphins are not produced, or they are at least they reduce their production, and now you got symptoms of withdrawal. So less pain relief, even natural pain relief, more pain because the receptors are not getting any pain relief, and that individual will go through extreme withdrawals. Okay, and this is related to some other things, so like neuronal pools, and facilitation. Okay, so these are pools or groups of interneurons. We already talked about this. Interneurons, typically, you can think of them as being in the brain. They're connecting neuron to neuron. They can form a group. Okay, so they can group up, form neuronal pools or groups of like nerves that are going to be stimulated at the same time. Okay. So they perform kind of a common function, and they will receive like input from one region of the body, and they might control one region of the body, or might be stimulated by one type of stimulus really well. Maybe some other stimuli will stimulate them, but just there's small patches within the brain that are going to be stimulated by like stimuli. And so they work together. Right? And facilitation is the impulse, okay, that is going to be released, okay, responds to a single impulse. Okay, so it's when you may have a neuron that causes a presynaptic neuron that causes a neuron to release more neurotransmitters in response to a single impulse. Okay, so the way this is it's like a summation. So if it continuously occurs, okay, if you have repeated impulses in the same region of the body, okay, then the presynaptic nerve will tend to cause that neuron to release more neurotransmitters to kind of speed up the process, to kind of make the neural impulse faster. Okay, and so this just increases the chance that the postsynaptic cell will reach the threshold. Right? And that's what that facilitation is. It's just repeated impulses that send out more neurotransmitters to increase the likelihood of that signal being carried further down the line. Okay? The issue with things like opioids, okay, is that you can overstimulate. You can have this occur, which then at first works great, okay, but then it detonates. Then it doesn't work as well, and you can have problems with the facilitation later on. Okay. Convergent, divergent. This is really has to do with what nerves are coming into the system. Convergent is one neuron that receives input from several neurons. Okay, and we'll look at a picture of this here shortly. Okay, and so normally what that is like I was talking about before, you can have sensory neurons from maybe vision, smell, ear, taste, touch, and they're all going to a single neuron. Okay, and so it's receiving several inputs from several receptors, okay, and then sending a signal. And it's allowing the nervous system to work more collectively. It can process that information, respond to that information, and it doesn't have to be a one-for-one kind of neuron connection. It's one to many, and so you can get a response that's much faster. Okay? And different sources receive different feedback. Divergence can happen this kind of same way, but it's in the opposite direction. So one neuron is sending multiple impulses to several neurons. So as a branching axon, okay, and it's attached or connected with multiple neurons. So you might have multiple neurons sending a signal back to a single neuron. Okay. And then you might have in the opposite direction a single neuron sending multiple impulses back to those regions. Might have a bunch of skeletal muscle, and so you have a lot of different skeletal muscles being excited at the same time in the same region. Okay, and so just what I said there, a single neuron in something like the central nervous system may activate several motor units in the skeletal muscle, or it could activate several glands potentially on maybe on like the surface of your skin. Maybe you have several sweat glands activated by a single nerve impulse, causing you to sweat across the entire surface, not just one neuron to one sweat gland kind of situation. Okay, instead it's divergent, one neuron, multiple glands or multiple muscles. So here's just a diagram of that. Okay, convergence, a bunch of neurons converging, right? Their axons and their synaptic knobs are converging on a single. Okay, so a single impulse. or you have one neuron sending out multiple impulses, okay, to multiple nerves, which then can send their own to whatever they're connected to. Could be another neuron, could be a muscle, could be a gland, on and on and on. All right, let's finish up by talking a little bit about drug addiction and kind of how that works when it comes to neurotransmitters and postsynaptic neurons. Okay? Often when, especially drugs that we might have that are not familiar to the body, or that maybe humans didn't evolve with, they can cause signals to be either stopped or enhanced. Okay, and so either you have this blockage of the signal or enhancement of the signal. So speeding it up or blocking it. Okay? And depending on what type of drug, an antagonistic, okay, drug is going to bind to the receptors and block the transmitter. So typically they're not going to send the signal. An agonistic, okay, is a drug that activates the receptor and causes action potential to occur, adding to the or adds to the binding of neurotransmitters. So it could open up the membrane, allow the membrane to be even more porous to the neurotransmitter, might entice the neurotransmitters to move faster, might allow for more binding that occurs at the neurotransmitter binding sites, these kind of different things. So 2 ways at which it works. Okay. When you repeatedly use certain drugs, you decrease their effect. You decrease their receptors, or you decrease how well they block things, or how well they activate receptors. Okay, so it causes the receptors to change. Sometimes by overexciting the receptors, by always using the receptors can be damaged and not replaced fast enough. Remember, these receptors are just proteins. These proteins are just amino acids chained together, okay? And they're sitting there, and every time they have to change shape and change form, they can get damaged, okay? And if you can't replenish them fast enough, okay, replenishing proteins happens because you use your DNA to code for messenger RNA, and that codes for amino acid chains. Then those proteins fold, and they're refolded and other things, and then they have to make it to wherever that protein might rest. Like in this case, typically the membrane of either a muscle or a neuron, things like that. Over time, if you continuously use them, okay, they get deadened. They lose their effectiveness. Okay? Amphetamines, okay, enhance neuroepinephrine activity, and so this often is one of those drugs that's really addictive and can alter receptors fairly permanently. Okay, and so what happens is they're enhancing neuroepinephrine activity. Okay, that obviously is beneficial to individuals because it elevates your alertness, your mood, it makes you more happy. Okay, makes you more active, awake, these kind of things. Nicotine, okay, same kind of thing. Okay, it's binding to the acetylcholine receptors. Okay, and so this allows for dopamine to be released, which kind of leads to that pleasurable feeling that individuals who smoke or use chewing tobacco or vape, etc., that has nicotine in it. Okay, same deal. So it's binding to the receptors, okay, which allows for dopamine to be released. In both cases, though, overexcitation of those receptors can be damaged, and over time, it takes more of these to excite to elevate the mood, to cause that pleasurable feeling in smoking, etc. Okay, and we'll look at some more examples as we progress.